Your body is destroying itself on purpose. Not by accident, not through neglect, on purpose, right now, inside every cell in your body. A clock is ticking, a biological countdown that has been running since the moment you were conceived. And at some point, without asking your permission, it will start winding down. This is aging, and for most of human history, nobody thought to ask why it happens. They didn't need to. You were born, you survived, you reproduced, and then you died. The average life expectancy for most of human prehistory hovered somewhere between 25 and 35 years. Not because humans couldn't live longer, but because infection, predation, injury, and starvation usually got there first. The few who did reach old age were treated as something close to miraculous. Repositories of accumulated knowledge in a world without books or writing. The memory of the group made flesh. But you are not your ancestors. You have vaccines. You have antibiotics. You have running water and grocery stores and emergency medicine. The external threats that killed most humans before they got old have, for many people, largely been managed. And now, for the first time in human history, we are dying of something that used to be rare enough to be almost invisible, old age itself. So what actually is it? The short answer is that aging is the accumulated failure of your body's repair systems. Your cells are constantly dividing, copying DNA, replacing worn-out tissue. But every time a cell divides, the copy is slightly imperfect. Errors accumulate, proteins misfold, cellular machinery gets gummed up with waste products. Your body has sophisticated mechanisms for catching and correcting most of these errors. But not all of them. Over decades, the uncorrected errors stack up, and the systems responsible for maintaining your tissues begin to malfunction. One of the most important mechanisms scientists have identified is the shortening of telomeres. Telomeres are protective caps on the ends of your chromosomes, the structures that carry your DNA, much like the plastic tips on the ends of shoelaces that stop them from fraying. Every time a cell divides, the telomere gets slightly shorter. When it gets short enough, the cell can no longer divide safely and enters a state called senescence, where it stops functioning normally but doesn't die. Senescent cells accumulate in aging tissues and release inflammatory signals that damage the tissue around them. Molecular biologist Elizabeth Blackburn and her collaborators won the Nobel Prize in Physiology or Medicine in 2009 partly for their work on telomeres and the enzyme telomerase, which can rebuild telomere length in certain cells, opening up one of the first genuine molecular windows onto the biology of aging. But telomeres are only one piece of a much larger puzzle. In 2013, a landmark paper by biogerontologist David Sinclair at Harvard Medical School and a team of collaborators proposed what they called the information theory of aging. The idea, put simply, is that aging is not just physical wear and tear but the gradual loss of biological information. Your genome, the full set of instructions for building and maintaining your body, remains largely intact into old age. What degrades is the epigenome, the layer of chemical switches and signals that tell your genes which ones to turn on and which to turn off. In which cell, at which time, over a lifetime, those switches get scrambled. Genes that should be silenced start firing. Genes that should be active go quiet. The result is cellular identity drift, where cells gradually forget what kind of cell they're supposed to be. And the tissues they make up begin to fail. This framing matters because it changes how you think about what aging is. If aging were purely mechanical damage, like rust on a machine, you would need to replace the damaged parts. But if aging is primarily an information loss problem, then the possibility opens up that the information might still be there, just scrambled, and that it might be possible, in principle, to restore it. Let's reconstruct what this looks like in your body. You're in your 20s, your cells divide accurately, your immune system clears senescent cells efficiently. Your mitochondria, the tiny power plants inside each cell, produce energy with minimal waste. Wounds heal fast. Inflammation resolves quickly. The epigenetic program your cells are running is clear and coherent. By your 40s, the errors have accumulated enough to become noticeable. Wound healing slows. Chronic low-level inflammation, sometimes called inflammaging, begins to build. Mitochondrial efficiency drops. Your immune system begins to lose the ability to distinguish self from threat. The epigenetic program is degrading at the edges, like a map left too long in the sun. By your 60s and 70s, those cascading failures accelerate. The senescent cells that your immune system used to clear are now accumulating faster than they can be removed. Tissues once capable of regenerating, like the lining of your blood vessels and the cartilage in your joints, are losing that capacity. The body is no longer effectively maintaining itself. All of this would have been essentially invisible through most of human evolution, because your ancestors were almost never alive to experience it. Here's the counterintuitive twist. From an evolutionary standpoint, aging is not a mistake. It is, in a brutal sense, the plan. Evolution selects for traits that help organisms survive and reproduce, not for traits that help them live indefinitely. Once you have passed reproductive age and raised your offspring to independence, natural selection has very little interest in keeping you in perfect condition. Biologist Peter Medawar articulated this insight in 1952, proposing that aging exists partly because the force of natural selection weakens with age. Genes that cause damage late in life, after reproduction, simply don't get weeded out the way early killing genes do. And some researchers have proposed a darker twist called antagonistic pleiotropy. The idea that some genes which cause damage late in life actually confer a survival advantage early on. Meaning evolution may have actively selected for the very mechanisms that eventually kill you. Your body was not built to last forever. It was built to last long enough. But here is where the story gets genuinely strange. In the last decade, scientists have begun to ask whether the aging process might be not just understood but reversed. And the evidence is suggesting, cautiously but unmistakably, that the answer might be yes. In 2006, Japanese stem cell researcher Shinya Yamanaka published a discovery that earned him the Nobel Prize, by introducing just 4 proteins, now called Yamanaka factors, into a mature adult cell. He could reprogram it back to a pluripotent state, essentially resetting its developmental clock to near zero. The cell forgot that it was old. This process, called cellular reprogramming, suggested for the first time that aging at the cellular level might not be a one-way street. Building on Yamanaka's work, David Sinclair's lab and others began experimenting with partial reprogramming, using the Yamanaka factors in carefully controlled pulses rather than continuous exposure, to reset the epigenetic age of cells without erasing their identity entirely. In 2020, Sinclair's team published a study in Nature showing that they could restore vision in old mice by partially reprogramming the cells of the retina, reversing age-related vision loss. In 2023, the same lab published further results suggesting that aging could be accelerated and reversed in living mice by manipulating the epigenetic program alone, without any genetic damage or repair. These results are extraordinary. They are also in mice. And the distance between a mouse and a human is vast. The honest scientific consensus right now is this. We know more about why we age than at any point in human history. We have identified several of the core molecular mechanisms. We have demonstrated that at least some of these mechanisms can be partially reversed in animal models. We do not yet know whether this translates to humans, how safe it would be to attempt, or what the long-term consequences of extended human lifespan might be. Companies and research institutions are pursuing clinical trials, but genuine anti-aging therapies for humans remain years or decades away, and the gap between promising mouse data and safe human application has humbled scientists many times before. What we can say is that aging is no longer simply assumed to be inevitable in the way it once was. For 300,000 years, your ancestors grew old and died before they had the chance to ask why. The question was irrelevant. Life was short, brutal, and full of more immediate threats. The body that carried you through those conditions was never designed with permanence in mind. Your body is destroying itself on purpose. But for the first time in human history, we are close enough to understanding exactly how it does it that the question of whether we can change the answer has become, cautiously and seriously, worth asking.